Hi, everyone. My name is Colin Sperling. I'm a fourth-year medical student, and we're excited to share with you our project, like you said, called Alteration of the Sphingosine 1-phosphate signaling pathway in the vaginal wall of women with pelvic organ prolapse. So, no disclosures. So, pelvic organ prolapse is a significant healthcare problem. It's estimated that up to 11% of women will suffer from this disorder at some point in their lives. And this can result in significant distress and diminished quality of life due to urinary dysfunction, bowel, and sexual dysfunction. While some of the risk factors for pelvic organ prolapse have been suggested, the mechanisms underlying the development of this disorder have not been elucidated. So for this project, we focused on the Sphingosine 1-phosphate signaling pathway, which is known to have countless metabolic functions in the human body, including the maintenance and contraction of smooth muscle. So while several previous studies have explored this pathway in several other tissues, our lab, to our knowledge, is the first to explore this pathway in vaginal smooth muscle. So our lab previously showed that Sphingosine 1-phosphate is formed from the conversion of sphingomyelin to sphingosine. In the presence of sphingosine kinase, sphingosine is phosphorylated to its bioactive form, sphingosine 1-phosphate, which we postulate could play a role in vaginal wall stability through cell survival and proliferation. In the presence of sphingosine phosphate phosphatase, this reaction is reversed, ultimately resulting in an accumulation of ceramide, which we believe could play a role in vaginal wall instability through cell cycle arrest and apoptosis. So basically our hypothesis is essentially that in dysregulation of this pathway, and specifically altered expression of sphingosine kinase, could ultimately result in vaginal wall instability and pelvic organ prolapse. So to carry out this experiment, we obtained anterior vaginal smooth muscle samples from two groups, women undergoing routine hysterectomy with no history of pelvic organ prolapse, and those with pelvic organ prolapse undergoing reconstructive surgery. We then conducted a molecular analysis utilizing Western blot with fluorescently labeled antibodies, which we then normalized to total protein. So after analyzing our data, what we found is that the expression of sphingosine kinase, which ran right around the expected molecular weight of about 43 kilodaltons, was roughly 46% lower in the pelvic organ prolapse samples compared with controls. So this suggests a quantitative deficiency, likely of some of the downstream molecules that could be implicated in this disorder. We also took a look at some other human tissue extracts and surprisingly found that the expression of sphingosine kinase was much stronger in our vaginal control smooth muscle sample, while relatively weak in both bladder and cardiac tissue. Equally as interesting, we also found that a rather robust expression of sphingosine kinase 1 in skeletal muscle, which we know is implicated in pelvic floor muscle and stability. We also analyzed the expression of sphingosine phosphate phosphatase expression, which ran at a slightly higher molecular weight of about 49 kilodaltons, and we found expression of this enzyme to be about 13% lower in pelvic organ prolapse samples compared with control. And so essentially the larger decrease in sphingosine kinase compared with sphingosine phosphate phosphatase suggests a larger sphingosine to sphingosine 1-phosphate ratio, thereby shifting the reaction towards decreased overall contractility and smooth muscle stability. So in order to demonstrate the presence of sphingosine kinase within these tissue samples, we carried out a DAPI analysis with a dual immunofluorescent staining. So as you can see on the left, the DAPI nuclear stain indicates the presence of cells in our sample. In the middle in green, we have a rabbit antibody specific for sphingosine kinase 1. And on the right, we have calponin, which is a specific marker for smooth muscle. When we did an overlay analysis, we see predominantly yellow staining, which indicates co-localization of the sphingosine 1-phosphate within our smooth muscle samples. So in conclusion, we demonstrated that sphingosine 1 kinase is the predominant sphingosine kinase found in these smooth muscle samples, and that levels of this enzyme were significantly decreased in women with pelvic organ prolapse. And this may suggest a possible preventative treatment strategy for patients at risk with this disorder. We had obvious limitations to the study. We only analyzed 5 control and 5 experimental samples, and we also only looked at a small part of a possibly larger pathway that could be implicated in this disorder. And in the future, we plan on analyzing more samples. Thanks very much.
Thank you.